The title of my presentation today is Microbiome-mediated plant soil feedbacks as a tool for resilient agriculture. Global agriculture faces severe challenges. By 2060, food production needs to rise by 50% to feed 10 billion people. Long-term monoculture, overuse of fertilizers and pesticides break beneficial plant-microbe partnerships, triggering soil degradation and frequent pathogen outbreaks. Plant soil feedback, or PSF, offers a viable solution to this agricultural crisis. First, PSF regulates rhizosphere microbial interactions that fundamentally determine crop yield. Second, host genes control root exudates to reshape soil microbiomes, enabling us to build positive PSF for nutrient cycling and pathogen suppression. Third, we combine microbiome-assisted breeding with adaptive diagnostic management to bridge lab research and field application. Fourth, meta-omics integrated with targeted agronomic and microbial interventions can reduce reliance on chemical agro-inputs. PSF serves as a theoretical framework to interpret plant-soil microbe interactions. It is a quantifiable and steerable ecological force shaping plant communities and biodiversity in natural ecosystems. There are two types of PSF. Negative PSF caused by allelochemicals and pathogens restrains crop growth. Positive PSF supported by beneficial microbes improves soil conditions, crop resistance and yields. Root exudates act as core biochemical signals mediating plant microbe communication. Key genetic loci including Feronia, NFR1, NFR5 and PHR1 have become targets for microbiome-assisted breeding. Flavonoids, strigolactones, organic acids and coumarins in root exudates recruit functional microbes to establish positive PSF. Native soil decomposers decompose organic carbon, boost nutrient cycling and improve soil structure. However, single microbial strains often fail to survive in complex field soil. We propose modular synthetic communities, or syncoms, combined with diversified cropping and conservation tillage to eliminate ecological barriers between lab and field for stable, positive PSF. Agronomic and resource management form the diagnostic system for PSF regulation. Conservation tillage stabilizes soil aggregates, rational rotation, intercropping and cover crops rebuild healthy soil microhabitats and dilute host-specific pathogens. Combined biological and physicochemical soil diagnosis helps cut pathogen loads and raise crop health and yields. Long-term crop domestication creates domestication syndrome and weakens microbial functional responses. Collaborative breeding is our core strategy, with three supporting approaches: microbiome-assisted selection, microbiome rewilding and vertical microbial transmission. We also built a three-tier diagnostic cycle including biotic assessment, abiotic monitoring and genotypic calibration to optimize field PSF status, demonstrated in Figure 5. We established a closed-loop pipeline: field sampling, multi-omics analysis, PSF status diagnosis, targeted intervention and long-term monitoring. Combining routine soil tests with metagenomics, metatranscriptomics and metabolomics generates quantifiable indicators to predict and adjust PSF, supporting precision ecological agriculture. First, PSF is predictable and quantifiable, and soil microbial legacies can be artificially managed. Second, meta-omics technologies are essential to realize precise PSF regulation. Third, our three-pillar framework focuses on synergistic integration rather than simple superposition of single methods. Fourth, our ultimate goal is ecology-driven sustainable agriculture for green and stable crop production.